Hello everyone, my name is Neil Patel, and today we'll be discussing purifying eukaryotic RNA polymerase II. This is from the University of Michigan from the course MCDB 427 Molecular Biology. As I previously mentioned, we'll be discussing eukaryotic RNA polymerase II and how the subunits were first discovered. RNA polymerase II is an enzyme that transcribes ribonucleic acids, also known as RNA, from deoxyribonucleic acids, DNA. One of the first experiments to look at the structures of the subunits of RNA polymerase II was done by Pierre Chambon and Rutter and their colleagues in 1971. However, they did not discover all the subunits at that time. I will not be discussing each individual subunit and their function, but if you want to pause the video, you can take a look at the different features of each of the subunits as listed below. One thing I do want to mention is that as you see here where it says RPB1, the gene names RPB2, RPB3, and so on, the RPB stands for RNA polymerase B, and the B stands for 2. So it would basically be RNA polymerase 2, and the 1, 2, and 3 of each, and continuing on to 4 through 12 are basically each of the genes. Now let's talk about the method. So one of the first things they did was to epitope tag RPB3. Just for clarification, an epitope is a part of an antigen that an antibody recognizes and attaches to. In this experiment, they engineered the epitope tag to RPB3. In order to do this, they had to 1. Know that the RPB3 is a part of RNA polymerase II, and 2. Know the sequence RPB3 in order to engineer the epitope tag to it. They added this gene to yeast cells lacking the functional RPB3 gene. They also added radioactive methionine or radioactive gamma phosphate. The general purpose of adding the radioactivity is to be able to visualize the proteins using such methods as autoradiography. To receive the RNA polymerase II, they lysed the cells later on and added an antibody that targeted the epitope. This method is known as co-immunoprecipitation, as we'll discuss on the next slide. Just a quick look in the figure, we can see the epitope tag is right here. And so we know that the protein is attached, the protein here is RPB3, and the rest of it is supposedly the RNA polymerase II complex. Let's move on to the co-immunoprecipitation. For co-immunoprecipitation, we first start off with the cell lysate. Cell lysate is basically everything that was within the cell, including DNA, RNAs, and protein, including a protein of interest, RNA polymerase II complex. We then incubate the cell lysate with antibody-coupled resin. This purple thing here is the resin, which is attached to the antibody. The antibody then looks for our epitope because that's what it's specific for. It can also potentially attach to other things in the cell, but it's highly unlikely since it's very specific, but it is a possibility. After a while, we spin down this mixture, and we'll have the antibody-coupled resin with whatever the antibody is attached to on the bottom of the mixture and in the tube, and on top of it will be the supernatant containing everything else within the cell. We then discard the supernatant, and then we'll just end up with our antibody-coupled resin, which is also attached to whatever the antibody is attached to. We then elute whatever the antibody is attached to, and have just hopefully the epitope tag RPB3 with the rest of the RNA polymerase complex. So going back to this image from before, here we could see through the picture our antibody. It doesn't show the bead, but theoretically it could have the bead, and you could do it that way as mentioned previously. And so our antibody recognizes our epitope, which is genetically engineered to the RPB3 protein. And then you have potentially the rest of the RNA polymerase II complex. Once you immunoprecipitate this with the antibody, you then subject it to a detergent like SDS, and then you'll have each of the individual peptides. Each individual peptide is supposed to signify whatever RPB or whatever RPB3 was attached to. So it should be each of the polymerase subunits. And then once we have this, we put it through a gel, and then each of the bands should correspond to each of the subunit each of the subunit proteins. So let's continue on to the results for our experiment. From the gel, we could see something that says epitope. Plus or minus. What does this mean? Minus means that there was no genetically engineered epitope tag to RPB3, whereas plus means there was a genetically engineered epitope tag to RPB3. The first two lanes contain radioactive methionine. This was basically used to see the presence of proteins. Lane 1 is a control lane. Let's start with that one. So there was no genetically engineered epitope tag to RPB3. So when we did add our antibody specific to the epitope, we were just trying to see if it attached to anything else in the cell that it was not the epitope tag. So if there's any non-specific binding. Sadly, we do see some bands, which means that our antibody is picking up some other things in the cell, but it'll be okay. In lane 2, however, we have our genetically engineered epitope tag to RPB3. And so each of these bands indicate something that was co-immunoprecipitated with RPB3 and the epitope tag. And so we could assume that these are the bands associated with RPB3 and which are also subunits of RNA polymerase II. So as you can see, we have band for RPB1, RPB2, RPB3, and it also says RPB4 here, although it's kind of light. The authors, let's just the authors on this one. Moving on to lanes 3 and 4. These lanes were with radioactive gamma phosphate. What's the purpose of that? That was basically to see if any of the subunits were phosphorylated. So let's start off with lane 4 in this case. Lane 4 had no genetically engineered epitope tag to RPB3. So this was just the antibody searching for anything that it picks up in the cell that is not the epitope. And so it was just trying to see if anything that it picked up in the cell, non-specifically the antibody, if that got phosphorylated. And as we see, it did not get phosphorylated. Anything that it picked up non-specifically. Lane 3, we see again we have an epitope tag that is genetically engineered to RPB3, and in this case we're seeing that anything attached to RPB3 in the epitope tag or the RPB3 itself, if it's phosphorylated or not. And we do see that RPB1 is phosphorylated as well as RPB6. So now let's move on to conclusions we can make from this particular experiment. So based on lane 2, we can see that there are at least 9 other polypeptides attached to RPB3. So we could see here, there is 1 right here, 2, 3 is the one that they're attached to, since this is the one that is epitope tag. So here's number 5, RPB5, 6, 7, although it's light, 8, 9, and 10. So they detected 9 other polypeptides other than RPB3 that were attached to it. So we could assume that RNA polymerase II has 10 subunits. From this data, we can see that potentially it has 10 subunits, or 10 subunits, 10 polypeptides that make it up. Another thing that we can see from this data is the subunits that get phosphorylated. So from this particular data, we can see that 2 proteins are phosphorylated. These include, from lanes 3, we can see that RPB1 gets phosphorylated, as well as RPB6. Although we cannot see it from data here, RPB2 actually gets phosphorylated too, but from this data, we have no way of actually knowing. So here is data from another experiment and some conclusions we can make from this experiment. So Roger Kornberg and colleagues conducted this experiment and basically similarly electrophoresed the subunits of RNA polymerase II. And here we could see something different. We could see actually there's 12 subunits. So based on all these bands, why didn't we see the other 2 bands? Well, that's because um, RPB11 and RPB12 co-electrophoresed. So here we can see that RPB11 and RPB9 co-electrophoresed together, which basically means that they had similar molecular weights, and so it's hard to distinguish between the two bands. They basically look like there was just one polypeptide at that place. But this they use a finer gel, and so because of this, the gel that they use, they were able to get this separation. And as you can see, there's two separate bands right there. Similarly, RPB10, RPB12 co-electrophoresed, and so there's actually two right there. So we can actually, from this data, we could say that there's 12 polypeptides that are associated with RNA polymerase II. And another thing that they have that we didn't see in the last experiment is a molecular weight standard. So with the molecular weight standard, we could see subunit sizes and make rough estimates on how big each of the subunits are. So I mentioned previously that RNA polymerase II subunits get phosphorylated. Well, at least some of them do. And these phosphorylations are very important, but we just don't have enough time to go through all of them and what they do and what they mean. Just one thing that I quickly want to mention is RPB1 phosphorylation. You're definitely going to see this in the future and the role that this plays. I just wanted to bring it up just so a quick little overview that these phosphorylations do play a role and they're not just there for fun. One of the things RPB1 phosphorylation does is play a key role in splicing, as you'll see later on in this course or in other science literature. This is just a citation of where the information came from. And yeah, I want you guys to have a great day and thank you for listening.